Bacteriology, a branch of microbiology dealing with the study of bacteria. The beginning of the bacteriology paralleled the development of the microscope. The first person to see microorganisms was probably the Dutch naturalist Antoine Leeuwenhoek. Antoine van Leeuwenhoek, who is in 1683 described some animal clues as they were then called in water, saliva, and other substances. These had been seen with simple lens, magnifying about 100-150 diameters. The organisms seem to correspond with some of the very large forms of bacteria. It's a bacteria, but he didn't know that it's a bacteria. He described it as animalcules. The very large forms of bacteria as we now recognized. He imagined something else. As late as the mid-19th century, bacteria were known only to the few experts in a few forms as curiosities of microscope, chiefly interesting for their minuteness and motility. It was aristocratic only. Modern understanding of the forms of bacteria dates from Ferdinand Cohn, a brilliant classification, the chiefly, but he shifted to the bacteria. While Cohn other advanced knowledge of the morphology of bacteria, other researchers such as Louis Pasteur and Robert Koch established the connection between bacteria and the process of fermentation and disease. In the process, discarding the theory of spontaneous generation and improving antisepsis in medical treatment. This man is very important in the history of bacteriology. From 1870 onward, he mostly studied bacteria. He published over 150 research reports during his life. The University of Priscillo became an innovative center for plant physiology and microbiology while he was there. His classification of bacteria into 4 groups based on the shape, spherical, short rods, threads, and spiral, is still in use today. Also, among other things, Cohn showed the bacillus can change from vegetative state to an endospore when conditions are hard. A heat-resistant spore of Bacillus subtilis caused a controversy over spontaneous generation. Cohn explained also why boiled infusion of hay or cheese could still get microbial growth. It was because they contain heat-resistant spores. He's a pioneer. He considered one of the pioneers of microbiology. The modern methods of bacteriology and bacteriological technique and their beginning in 1885 with the introduction of the use of stains. Koch, by the discovery of the methods of separating mixture of organism on plate of nutrient media solidified with gelatin or agar. This man also a pioneer. Robert Koch discovered the TB, anthracis, and many other bacteria. He make a tuberculin and describe it as a treatment. Bacteria is a singular, and the bacteria is the plural of singular bacterium. Bacterium is the single, bacteria is the plural. Any of group of microscopic single-celled organisms that live in enormous number in almost every environment on Earth, from deep sea vents to deep below Earth surface to the digestive tract of the human. It's all over. Bacteria is in the singular bacterium. single cell microorganism found everywhere in the environment, in the sea, in our digestive tract, everywhere. Important discoveries came in 1881 when Pasteur succeeded in immunizing animals against two diseases caused by the bacteria. His research led to the study of disease prevention and the treatment of the disease by vaccine and immune serum. It's a branch, the immunology, the branch of the medicine. Consider the immune system and its function. Other scientists recognize the importance of bacteria in agriculture and the dairy industry. Bacteriological study subsequently developed a number of specializations, among which are agricultural or soil bacteriology, clinical diagnostic bacteriology, industrial bacteriology, marine bacteriology, public health bacteriology, sanitary or hygienic bacteriology, and systematic bacteriology, which deals with taxonomy. The bacteria lack a membrane bound nucleus. Inside there is no membrane bound nucleus and other internal structure, and are therefore ranked among the unicellular life form called prokaryotes. And this is the morphology of the cell, the bacterial cell. The topics of general bacteriology include bacterial morphology, structure of the bacterial cell anatomy, bacterial physiology. Bacterial in the physiology we study growth, bacterial nutrition, bacterial product, the bacterial genetic and how it's resist our antibiotics, and how can transfer some of its genes from one to another, host parasite relationship, commensalism or parasitism, the antimicrobial and how it's affected the bacteria. It's a product of the bacteria to defend itself, and how we use this antimicrobial to fight bacteria, and how it gets resistance from these antibiotics. The systematic bacteriology is a branch of bacteriology concerned with nomenclature and classification, the taxonomy. We put the bacteria in families, in genuses, in subspecies, species, subspecies, to study it simply. Bergey's Manual of Systemic Bacteriology is a manual referring to the taxonomy of prokaryotic bacteria. It was prepared by the American bacteriologist David Hendrik Bergey in 1923. It's a manual that deals with the identification of bacteria. From 1984, Bergey's manual was renamed Bergey's Manual of Systematic Bacteriology. Is being published in separate volumes. This manual includes 35 sections based on the character like general shapes, morphology, gram stain, presence of endospore, motility, oxygen relationship, mode of energy protection. Thank you.